So as I said earlier, we have to, on Thursday and Friday, we have this split schedule. We have no time to play around. You have to come in and get this right. So we're going to look at this biomolecule. This is a fill-in. Obviously on the test, it'll be multiple choice, but you can expect the same content questions. You have 4 biomolecules, either they're proteins, lipids, carbohydrates, or nucleic acids. The only choices you have. The first one here says used by the body for long-term energy storage. At this point, you should know it's lipids. Contains nitrogen. The only one of these that we talked about as having nitrogen is the one with an amino group or amino acid group monomer. We know that's protein. 2 types of monosaccharides and disaccharides. You either have monosaccharides or you have disaccharides. Yes, they're carbohydrates. This should have been easy, done. I should have been able to, if this was your test, you should have been, thank God. Simple fill in with the answers spelled correctly above you. I'm just going to put carbs. Used by the body for a quick energy source. Quick. The key word here is quick. Notice that the other key word up here, this one says quick. This one up here says long term. You see the difference. So long term, it's lipids. What's the short term? Carbs is correct. And again, carbohydrates. I'm writing quickly, so it's going to be messy. You have the right spelling and words up here. Contains phosphorus. The only biomolecule that contains phosphorus. Nucleic acids, correct. You know what? I'm just going to, I have to do it short. A, B, C, or D. Okay? Because I can't keep writing this in. We have no time. Used by the body for insulation to keep yourself warm. Lipids is correct. So that's B. An example of an enzyme is an enzyme. It is, protein is correct. I have to give you a warning. Sometimes it is nucleic acids, but we'll talk about that as we move along. Tuna and other meats contain much of this. Protein is correct. Pasta contains much of this. Carbohydrates. Stores information in your cells in the form of a code. Nucleic acids. Building block in an amino acid. The building block is an amino acid. Protein's correct. Butter contains much of this. Lipids. Look how, see? Easy, yes? Use the exoskeleton in insects. I actually told you that. Carb. C. It's chitin. Chitin. Let me write that in because you will get a question on this. I'll make sure of it. Chitin. It's a carb. It's a structural. Let me erase this and say there are 2 forms that we talked about: structural polysaccharides. What are polysaccharides? What are polysaccharides? We have monosaccharide. What does mono mean? 1. We have disaccharide, which is what? in your study guide, it talks about these. An example, monosaccharide is glucose. So if you've been studying, if you've been following what I told you to do, you should know this. This you should have known already. This is showing you your weaknesses, right? So you have a weekend to build yourself up. Monosaccharide would be glucose. Another one would be what? Ribose. Those are the two I told you need to know. Two of the most important ones you got there. Disaccharide, the example you have in your study guide is sucrose. And you can look at the study guide and take a look at it closely. You'll see there's two of them. A disaccharide, di means two. 1 and 2. Polysaccharide means many. So many, many more than 2. So some polysaccharides, their starch is 1, but starch is not a structural one. Energy storage, polysaccharide used for energy storage is starch in plants and glycogen in animals, in muscle.
Isn't there cellulose too?
There is, in muscle and in liver. But understand, that's where you find it. You find this glycogen in the muscle and liver of animals. Starch is only found in plants. All right, very big difference. That's for energy storage. Now, structure, meaning to hold yourself together, to give yourself a skeleton, for instance, an exoskeleton, meaning the skeleton on the outside, like insects and beetles and arachnids, correct? Crabs, you know they have exoskeletons, correct? Yes or no? No, you did not know that. Okay. You know you have a skeleton, correct? That's on the inside of your body. You don't have a skeleton on the outside? Huh? Yeah. There's something called an exoskeleton, where things have a skeleton on the outside. Have you ever eaten crab or lobster? What do they have on the outside? Shell. Do they have any bones on the inside? No. Because their bones are on the outside. That's why we call them exoskeletons. They don't have bones. They have an exoskeleton. Well, insects have an exoskeleton made of structural polysaccharide called chitin. Have you ever felt a crunch after you killed an insect or as you're killing one? That crunch is the exoskeleton breaking. Chitin is what it is. It's made of sugar. That exoskeleton is made of sugar. Another polysaccharide that's structural, somebody said it already, is cellulose. Those are the two structural polysaccharides. So you should know that. That should be a review. Let's go back. Used in the exoskeleton of insects, chitin. Probably misspelling. Someone want to look up the spelling really quickly so I don't leave an error on the notes here. Help transports oxygen. You probably don't know this. It's a protein. It's A. That is called hemoglobin. Yeah. And you said that and I put an I there. I think it is still an I here, hemoglobin. All right, so this is chitin is with an I. I did it again. Hemoglobin is the protein that carries oxygen in your blood, makes your blood red. Can be used in murder trials. Nucleic acids. I don't like that one because a lot of stuff can be used in murder trials, but that's fine. Label the following compounds as organic and inorganic. Let's be clear about something. It's in your book, but let me make sure you understand. Organic does not mean homegrown. In the sciences, organic means it has carbon. Carbon-based. All right, carbon-based. Comes from life. So usually carbon and hydrogen. Carbon, oxygen, and hydrogen. At least. Okay? Things that come from living systems. So carbon dioxide, for instance, is not organic. It's inorganic. So this is not organic. This is what? C6H12O6. It's organic. It's also sugar, but you should know that, by the way, but moving on. Water, is that organic or inorganic? It's inorganic because it has no carbon. NH3, ammonia. Is there carbon? No, inorganic. It's funny because this is made by your body. This is the basis of pee. When you're pee, this is the waste. This is what the weight, that stuff that smells in urine, that's the waste of your body. That's the actual waste from inside your body. Poop never was in your body. So when you poop, that's never been inside you. It's just been in your intestines, in your mouth, your stomach, and your intestines. That's never been inside your body. Your urine is what's been in your body. You're putting it out. CH4. Organic is correct. And H2O2? Inorganic. It's tricky, to be honest, organic, inorganic, but the bottom line is carbon, probably carbon and oxygen and hydrogen, you're pretty sure it's organic. Matching. Simplest carbohydrate is which one? 26. So simplest carbohydrate A is monosaccharide. The substance that contains carbon. That's right, organic. CO2 being the exception. Protein that speeds up chemical reaction. You should have read that in your book, but it's not going to be on a test. It is called a what if you've read your book? Enzyme is correct. Contains double bonds, liquid at room temperature. Unsaturated is correct. Made up of two simple sugars bonded together. Two simple sugars bonded together. Simple sugars. Sugars are also known as saccharide. Two. Poly is many. Saccharide is sugar, but poly means many. This is how many? Two. What's another prefix for two? Di. So it's E. Contains single bonds and solid at room temperature, saturated fats. So you look at meat, you look at butter, they're saturated fats. Examples include glycogen and cellulose. We literally just went over it. And it's the only one left. Polysaccharides, G. Cellulose and glycogen. Oh, by the way, yeah, glycogen's there. What is going on? Why are you getting up? Okay, you raise your hand and you say, Mr. Mendoza, it's time to go. Sit down. Let's get in that habit. Let's get in the habit. Just sit down. Let's sit down. Let's sit down. Raise your hand and say it. Go ahead. Yes, sir. Oh, is it? Oh, wonderful. Okay. We're going to be back at lunch. Hurry up back. We're after lunch so we can finish this. So that's it. Carbon-based, organic. Organic means it's carbon-based. You have to be careful, though. It's not always that easy. Like carbon dioxide is not organic. It's inorganic. The rest of these, I think it made sense to everybody. I'm moving on. Says Betsy and Sarah performed the same lab testing for fats, proteins, and carbohydrates that we did in class. We didn't do it in class. I skipped that one. We can use things called indicators. Use their data to answer the following questions. So there's 3 tests. The brown paper test, you know it. You know what the brown paper test is for fats. What is the brown bag test for fats? What is it? You know it. You've never seen a brown paper bag? Never seen one? Okay, you have. Have you ever put something greasy in a brown paper bag? Yeah. Chicken or acapulco or something. Yeah. What empanadilla, whatever. What happens to the paper bag? Yeah. Yeah, this grease seeps through, it gets shiny and slippery. That's the brown. That is a test to see if you have fats. Water doesn't do that. Other things don't do that. Is that clear? So that's a brown paper test to see if something's fat. You can take a brown paper bag, put it in a brown paper bag. If it's a fat, it'll seep through. The paper bag will absorb it. It'll get shiny. I hope some of you are staring into space. I hope you're paying attention. Iodine test. You can take iodine, put a drop in a solution, a drop of the solution onto anything that has starch, and it will turn black. It will turn black. It's an indicator. A dark brown. Benedict's test for sugar. And Biarrit's test for protein. We're not going to go into these tests. You don't have to do them. But I just want you to know that these tests help us identify if there's sugar in something, if there's fat in something, if there's protein in something. We call them indicators. Do you understand what I'm saying? Okay, so you don't have to do this. So you should be able to define starch. What is starch? Yeah. What is it? Plants have it. That's correct. Made by plants. What else do you know about starch? It's a polysaccharide. In other words, as many monosaccharides put together. It's stored in the roots. This is all in your notes. If you have taken good notes. If you have taken good notes, it's in your notes already. I know a lot of you have not taken good notes. We're still working on it. But I don't know what else to tell you. I told you in class, you should know it. Roots, it's in roots. It's a polysaccharide, and they use it for food storage of energy. Glycogen is what? What's glycogen? A lot of quiet, even though we just answered it on the previous page, by the way. We answered all this in the previous page. What's glycogen? Animals. And what? And muscles is correct. And liver is correct in humans. And what do we do with it? Why do we make it? Why do we have glycogen in our liver and our muscles? To store it. To store energy. Yeah. To store energy in the form of sugar. So it's a polysaccharide made for storing energy. What about cellulose?
Plants use it for structure.
That's right. Plants use it for structure. There's two of those documents in your assignment. What are the building blocks of lipids? They don't have they're not polymers, but there are some building blocks we've discussed. Glycerol. If you don't know, I see a lot of blank faces. It's in your notes. Glycerol, fatty acids. Proteins are what? Building blocks of proteins are what? Amino acids. I don't know why this thing's acting up. I can't zoom out because I'm writing the answer in the document. Is a pencil almost dead? Is that what's happening? Pencils. I don't know what's going on. Having trouble here, guys. Give me a second. Amino acids. Everybody should know that at this point. Carbohydrates is the building block of carbohydrates. Say what? That's right. That's exactly right. Monosaccharides. And nucleic acids, the building block. The only thing you have to know about nucleic acids, nucleotide. You have to know a nucleotide is the building block and that the components of a nucleotide, the ribose sugar, deoxyribose sugar, the phosphate, and the nitrogenous base. What is an enzyme? It's a protein. That's right. That increase speed of reaction. Again, right out of your chapter reading. Which biomolecule is it? Well, we already said it's a protein. Use the following elements. Schnapps. There's sulfur as well. I don't like schnapps. Answer the following questions below. Which element make up a carbohydrate? CHO. Specifically 1 to 2 to 1 ratio. Remember that. That's going to help you a lot. That's important because of the next one. The next one's a lipid. What is it made of? Also CHO, but mostly what? What is it mostly made of? C and H. Lipids are made mostly of carbon and hydrogen. Zoom in, zoom out. You guys, can you see that? CHO. Where lipids, it's mostly C and H. What about a protein? What elements are proteins made out of? C. I have to zoom out again to write. What is going on? Wow. CHO and N. Notice, please notice that this is not in lipids or in carbs. There is no nitrogen, that's an N. There is no nitrogen in carbs or lipids, for the most part. Elements made of nucleic acid, what do they have? CHON, except they also have a P. So that's how you're going to tell the difference on a test, like the one on Monday. If it's CHO, 1 to 2 to 1 ratio, it's a carb. If it's a CH, mostly carbon and hydrogen, there's some oxygen in it, it's a lipid. CHON, there's nitrogen in it, but no phosphorus, it's probably a protein. If there's CHON and P, a phosphorus atom or phosphate group, then it's a nucleic acid. Are we clear? All right. Please know that. And there's an experiment here. It's asking the independent dependent variable. You will get a question like this on the test. For the record, Carmen is a long-distance runner and wants to determine which food will give her the most energy. She monitors her times over the 3-week period. For every race she runs 1 mile at the same time of day in the same weather conditions. Her data is shown below. Same mile, same conditions. These are her times. When she eats lipids, she only changes what? What's the only thing she's changing? This is the food she ate. What changes? The time. So which is the independent variable? The food. Which is the dependent variable? All right? Right? Easy. Easy. What's the independent variable? The food. What's the dependent variable? The time. List 3 constants in the experiment. What's a constant? Exactly. Conditions, time of day. What's the only, is this a good experiment? No. Why not? There's something missing. How do you know what does she run normally when she's eating all the food normally? Her control group. You need to have something to compare it to. These are the ones you changed. She should have given us a time where she runs it under all these same circumstances except she's eating normally. Right? A good balanced meal. So that would be her control group. So she's missing a control group. Are we good with that? All right, so that's it. I really feel you guys did a good job learning that material. I'm hoping that you're going to get at least 100% on those. Back and look. You just said it. Look at it. It says what? Made mostly of carbon and hydrogen. Made mostly of carbon and hydrogen. Lipids. If for every carbon you had 1 oxygen, then you would think section of the test. Now let's look more at biomolecules. This is the other document. Please don't lay your head down. Please don't lay your head down while we're going over a review. Don't do that. You should be looking at the screen, copying stuff down. Giving me a dirty look isn't going to help you. Lift up your head and look at the screen. Take notes. Notebook open. Young lady, sit up.
Bro, I can see the screen.
No, sit up. Take notes. So remembering when 2 lines connecting a diagram such as this, there's a carbon. So you see 2 lines here connecting. That's a carbon, correct? That's a carbon, that's a carbon, that's a carbon. So you can't see what I'm looking at if you're not looking at the screen. Absolutely 0 on this. All right, why don't you get your stuff, go outside. I'll call your mom. We'll figure out why you don't care. So this is a carbon. Take all your stuff. Just work on this outside. Take a couple stools. Okay, very good. We'll make sure to communicate that. Carbon, 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 carbon. Where you don't see any other element listed, how many bonds are in each one? 4 bonds for each carbon. So if there's one, this carbon's bonded to this carbon's bonded to this carbon, that's 2. How many more bonds does this carbon have? 2. What's it connected to if it's not showing anything? Hydrogen. They don't show the hydrogens. All the missing bonds are connected to hydrogen. Is that clear? Mr. Mendoza's room. Mr. Mendoza speaking. May I help you? There was a power outage at the microwave. I didn't send anyone to the dining hall. I don't know why that would be an issue for them. I don't know what's going on. They did. All right. Yeah, the power. Something happened. I don't know. I think a circuit breaker broke or tripped at the custodial level. That's all I can. I'm not sure what else. You're welcome. Bye. All right. So when we're talking about these bonds, you're looking at this complicated molecule. By the way, this molecule is what we call a steroid. When you think about steroids, this is what they look like in general, a bunch of rings, usually 4 or 5 rings of carbons connected to one another. Which of these is it? What's it mostly made of? Carbon and hydrogen, right? So what is it? No. It's not. What is it? Go back at your previous notes. What is it mostly made of? Carbon and hydrogen. Should we go back? Let's go. Then you would think carbohydrate. But look, there's a carbon, there's no oxygen connected to it. Look at all these carbons without oxygen. This is obviously a lipid. This is an application of a simple idea. If you have carbon and hydrogen and no oxygens, mostly carbon and hydrogen, it's called a lipid. So the answer here is lipid. Are we clear? I guarantee you're going to get a question like this on a test. It won't be this exact one. This molecule is made of glucose. Glucose monomers. What is it? It's a polymer. It's a polymer. Now, to be clear, if it was just made of 2, it would be a dimer, just the number 2. If it was more than 2, greater than 2, an oligomer is greater than 2, but less than poly. Man, that is too big. But less than poly. Is that clear? Yes? But you don't have to worry about oligomer. You won't see it in here. Just to let you know, you might see it in a book. You might see it when you do research over the summer. You might see any kind of stuff like that. If you're going to do an internship over the summer at Cleveland Clinic UH in a lab, you're likely going to see these terms. It's obviously not it's actually a polymer is correct. What else is it? B? D as in dog? Ah, yeah. It is. It's polysaccharide as well. It's a polymer and a polysaccharide. So the answer is actually E. E is the actual answer to the question because a polysaccharide is a polymer. You're walking down the tough streets of New Jersey, and suddenly you feel the need to run away from someone who's accosting you, who's chasing you, trying to kill you, hurt you, whatever. Your muscles keep a form of energy stored just for this emergency. What macromolecule stores energy in the muscles and liver? Glycogen. So you've gone all day without eating. You're walking home. Somebody's chasing you. You run. Where'd you get that energy? You didn't eat. You had it stored in your muscles and your liver. You release it quickly in an emergency. You're ready to go. And you can't depend on just being in the liver. You need in the muscles as well because your muscles need to react. Even if you're hungry, you have to have that energy stored somewhere. That's why people slow down when they haven't eaten for a few days. They're running out of energy. All right, let's take a look at this. Gives rigidity to cell membrane. You wouldn't know that. You can not worry about that one. Not worry about that one. That'll be on the next test. Keep that question in mind, though, because when we go over it next week, you know what kind of question you're going to get. The molecule below is a what? What is it? Quick. Unsaturated fatty acids. Should know that without too much trouble. Should be able to see here, double bond, unsaturated. Long chain of carbon. Table sugar is what? It's sucrose. That was right out of your study guide. It literally says, we said it was table sugar, sucrose is right in there. Read it, study it. You were given that 3 weeks ago. Disaccharide is formed when two monosaccharides are joined together to form, are joined together by what? How do we join two monosaccharides together? A synthesis reaction is correct. Specifically, what kind of synthesis? Dehydration synthesis is correct. So it is this. Hydrolysis does what? What does hydrolysis do?
It puts in water to break it.
That's right. So hydrolysis does the opposite. Hydrolysis does the opposite. It doesn't put it together. Lysis means break apart. Lysis means break apart. So don't confuse those two. It's synthesis. Synthesis means make. Dehydration synthesis. You should know what it looks like. Go back and review the notes. Class notes. You're not going to find it in your book. That's not in your book. That's right out of your class notes. The main source of energy for cells is what? Which of these? Glucose is correct. Whatever we eat, we want to turn it to glucose so that we can digest it correctly. Your body runs on glucose. We ready to go on? Mr. Jolly has just been to his doctor to discuss the results of his recent physical. His doctor told him that his blood bad triglycerides and cholesterol are too high, and that he is beginning to develop heart disease. Since he has not completely developed heart disease, there's still time. Mr. Jolly to change his diet and lifestyle so that he does not develop heart disease. Given his new health news, what would be the best dinner for Mr. Jolly to eat tonight? What's bad? Remember we talked about this. When you have a triglyceride, three carbon molecule that you have attached fatty acids to, the wiggly line is a chain of carbon and hydrogen. What's a bad one versus a good one? That's one triglyceride. Well, the question is, what's a bad versus a good? What kind of fat is good versus what kind of fat is, quote unquote, bad? That's right. Did you hear what she said? I told you in class, saturated fatty acids, not as good. You still need them. They're all single bonds versus unsaturated. Newer research shows that we still need saturated fatty acids. We just need less of it because it's not as healthy for you as unsaturated. All right, unsaturated means double bonds. I sketched that out. You can go look at the notes to discuss what's saturated versus unsaturated. So you want to eat something with unsaturated fatty acids. That means something is cooked in oil that in lipids that are solid or liquid at room temperature. Liquid. The solid is stuff like lard and butter. Lard and butter, animal fat is going to be saturated fatty acids. You want to avoid pizza and donuts because pizza has cheese and donuts fried. So the rest of these have a lot of carbs. You probably want to avoid cheeseburgers, French fries, and chocolate ice cream as well. I said avoid. Did I say get rid of? No. Say avoid. Don't eat 3 cheeseburgers a day. Don't eat a cheeseburger every day. That's what they're talking about here. Reduce the amount you eat of these fried processed foods. I don't think that's hard. Nobody says you can't eat them. But eating a donut every day for breakfast, probably not a good idea. Mr. Jolly had put himself at risk for heart disease due to his previous eating habits. Which of the following is most likely describes the eating habits that led Mr. Oh wait, was this the one that he? Oh, the best dinner. I'm sorry, I'm circling the wrong answers here. The best dinner is which one? D. Whole wheat pastas, olive oil, veggies, and tofu, all of which have unsaturated fatty acids. You have to go somewhere? Where are you going? What is this? You have to get a pass from the office. Your parents have to call the office to have them excuse you. I can't just let you go home. Have them call the office. Wait, sit down. Call your mother and tell them to call the office. You should have taken, what is this? Who is this addressed to? Who does that say? Does that say Mr. Mendoza? Oh, they signed it. Well, you didn't tell me that she signed it. You just hand me something that says Miss Prez. I don't know what that means. People handing me stuff at the last minute. I don't know what's going on. Mr. Jolly had put himself at risk for heart disease due to his previous eating habits. Which of the following most likely describes the eating habits that Mr. Jolly had? High-risk heart disease. A diet, what? A is correct. Not hard, correct? Yes. Easy. Potatoes have a large amount of what? Starch. Boom. Done. Lard is pig fat, animal fat. People often use it to make pastries. Tastes delicious. Bacon grease and lard. You probably ate it in gravy, in biscuits. They use it to cook biscuits. It's delicious. And those of you that say ill, but you eat meat, I don't know what you're talking about. You eat this all the time. It's just in less quantity. So it's solid at room temperature, and it's really good for making pastries flaky. So really, anytime you eat any kind of pastry that has flaky stuff, like do you know what flaky means? Yeah. All right. So any pastry that's flaky, that's in layers, like strudel, it has a lot of butter in it. Has to have saturated fats, so that it cooks up and it basically fries each layer separately. Tastes delicious, great texture, horrible for you. All right. Lard is a what? Saturated fatty acids in a triglyceride form, obviously. During photosynthesis, plants capture sunlight energy to make glucose. Then they are able to store the glucose in what macromolecule? Starch. They store it if they're storing sugar. Of course, if they're going to build their bodies, they're going to use cellulose. It's the same macromolecule. Can I help you?
It's Marbury asked for me to watch this out here room.
All right, no problem. Which food below, when consumed in excess over time, would be most likely the cause of heart disease? I would say butter. Why not peanut butter? What's peanut made out of? Peanut butter made out of? Peanut oil. We make peanut butter a solid at room temperature, which is a good guess, a peanut butter, because you're thinking it's solid at room temperature. But we make it that way. Normally, the peanut oil is liquid at room temperature.
So sugar is not healthy?
Sugar cane, what's sugar? Is it an oil at all? No. It's not a lipid at all. It's a carbohydrate. It's still bad for you. There's everything in excess is bad for you. You have to eat sugar. You just don't want to eat sugar cane. You don't want to eat pure glucose or sucrose. You don't want to eat table sugar. And we'll talk about why later when we talk about diabetes. 15. A plant living in a dry area will likely have a substance that helps minimize water loss from the leaves. There'll be something outside the leaves. What is that substance? It is a cuticle, but what's the substance on the cuticle? Yeah, it's going to have all three of them. It's going to have a wax. The wax is a lipid, and it's called the place is called the cuticle. So it's all three of these. The answer is E. In fact, we call them wax. You never seen leaves that like a cactus leaves, aloe vera. Their leaves are thick and they're kind of shiny in the sun, in the light. That's because they have a wax coating. They protect from water loss. When you're worried about getting dry lips, what do? You put wax on your lips. Burt's Bees wax, right? Some people call it. Some people have Carmax, which is a wax, right? All you're doing is putting a wax on your lip, a lipid, a fat on your lip to keep your skin from your lips from drying out. When you put lotion, what are you slathering on your skin? What's the lotion made of? Not wax. It's a wax is a thicker lipid. It's a lipid. It's a lipid. It's fat. All you're doing is taking fat and rubbing it on yourself. In the old days, before we had processed stuff, people would use olive oil. They would use all kinds of oils to hydrate, to keep their skin moist in dry areas. Fat, animal fat. They just would lay it up on themselves. Hey, when your skin's drying up, you live in a desert, it's either that or be ashy and itchy and crack. Their skin would crack open. Not good. If you have consistent issues with bowel constipation, which of the following should you increase in your diet? You don't know this, but it would be cellulose. Cellulose is the undigestible material in plants. When you eat leaves like lettuce, like any plants, you ever eat a carrot and you went and it crunched? That you're biting through cellulose. It's the same stuff you're sitting on, right? There's just less of it. The image below shows a buildup of plaque on the inside of your artery wall. Which of the following dietary macromolecules is most likely to cause a type of plaque buildup? Not unsaturated. Saturated. I keep saying it's saturated fatty acids. It's not going to be earwax unless you're eating earwax, which is really weird.
It's B?
It's B. It could be saturated. You could do B and I guess you can do B and C if you'd like. No, it's not B at all. I'm sorry. No, I'm right. It is B. For sure, let's go with the best answer. So there's only one answer. It's C. Hey, 100 questions. I had problems with one, and you're going to jump all over me? So I don't know how many questions are in here. Macromolecules depicted in this image. It's a bonus question, so you can answer that one on your own. You can answer that one on your own. It's a bonus question. Which of the following is the long-term energy storage of both plants and animals? Both plants and animals. Both cellulose is found in where? Plants, not animals. Glycogen is found in animals, not plants. Starch is only in plants. Chitin is insects. So, triglycerides. I don't really think that's very accurate, but whatever. I'm not even sure how do you long-term storage of triglycerides? In adipose tissue, that's true. Triglycerides in fat, right? The fat around my belly is triglycerides inside cells called adipose. Their job is to store fat, to keep me warm in the winter. It keeps me protected from physical injury. If somebody punches me in the stomach, I'm not going to get damage to my kidneys or my lungs, so they'll hit the fat. So it actually acts as an insulator. All right? Too much of it obviously causes disease. So molecule below is a what? Are they saturated or unsaturated? Saturated. They're lined up. See how they're lined up together? They stack up. If they're stacking up, it's going to be a solid. It's saturated. If it's saturated, it's not a solid at room temperature. That was a bonus, but moving on. Macromolecule below, which consists of chain of glucose stacked on top of one another, is called a what? This is extra credit. You'd have to look it up. I'm going to let you look it up. I know what it is because I looked it up. Lipid molecule transmissive. Again, another bonus question. I'm going to let you look it up if you want. You don't have to answer them. They're not going to be on the test. In fact, most of those with health ones are not going to be on the test. I just wanted you to get an idea of what we're talking about, put it in context. You might get one, but I doubt it. We only have 20. Remember, we only have 20 to 30 questions on this test. I don't have because it's during a normal school day, not this extended time. Is that clear? So and you think, cool, only 20 questions. That means each one's worth more, right? It's 32. I'm debating whether to keep it 32 or to bring it back down. I'm debating. I'm asking some of my other upperclassmen to look at it and see if we should reduce the number. Information macromolecule that stores information about how to run a cell is, where do you store the information on how to run a cell? It's in your study guide on the biomolecule study fact sheet. DNA, nucleic acids in general. Proteins are made up of chains of amino acids. All right. How many different amino acids are there? 20. Change in pH is able to cause protein denaturation because you won't know that. That's extra credit. Bonus points. Bonus. Answer it and you get bonus points. You can come see me after school, before school, if you want an answer to that. We'll cover it later in the year, next week actually, or week after. Amino acids are linked together via what kind of bond? Peptide bond. What type of reaction links monomers together to form a polymer? Synthesis, dehydration synthesis specifically. Which of the following is non-organic compound? H2O, water. Which of the following represents the correct ranking in terms of smallest to largest? So you got carbon atom, macromolecule, polymer, monomer. It's not C. E. Carbon, atom, monomer, polymer, macromolecule. That is E. That is the correct answer. Pay attention, guys. I warn you, this stuff is going to get overwhelming if you're not taking things one at a time. You are a scientist at a prestigious university. You are studying the effects of various temperatures on the function of particular fictitious cellular protein, tox eliminator. The function of the protein in a cell is to break apart and eliminate toxins that invade the cell. The following chart depicts the experimental setup. You take a normal cell temperature, 2 milliliters of toxin, 2 milliliters of the toxin eliminator. You do the same at each temperature. So this is what you're doing. You have 4 groups, right? This is normal. What would you think you're calling this one? Body temperature, normal cell temperature. What would you call this? Yeah, that's right. That's your control group. That's right. I like how you said it after she said control. I hope it shows up on the audio. So this is your control group. Then you have 3 experimental groups. I haven't even read further, and I know I'm going to start to break it down this way. Which of these is the independent variable? The one you're changing. Temperature. And which is the dependent variable? That's right. For your data, you measure the amount of toxin still left in each test tube 10 minutes after you have mixed the test tube ingredients. You have written down your results, but you have been a sloppy scientist. Gee, I wonder where we've seen that before. Wonder where we've seen that before. All these dead plants, or plants that none didn't survive, that were meant to. You mixed up the tubes, and now you don't know which measurement goes in which tube. This is your following data. This is the amount of toxin in the tube. So you have, this is what you have, this amount of toxin. Which piece of data most likely corresponds to tube D at a high temperature? Not, this is a different set of questions. So you wouldn't be able to answer these. I thought it was something else. I apologize. We can just get rid of all these questions for now. We will come back to this, though, after next week. As we go along next week, make sure you look at this experiment. Think about this experiment, because you will see them. And that's what your test is going to be like, something like that, except the stuff that I X'd out. 20 to 32 questions, depending if there's fill-in, probably something in between. All right, yes. So your job is sit down, everyone, except the one person's going to the bathroom. So your job, yeah.
Is this we write this on paper?
What?
The test.
The test? Will the test on Monday be on paper? Is that what you're asking? Yes. Why are people upset? I know why. They're frustrated, right? They're frustrated. Amount of effort you put in, amount of times you studied, how much time. Imagine if the test would have been today. Imagine if the test would have turned in today. A lot of people would have passed with an A, guaranteed. In this room, I know of at least 5 people. You know how I know? Because at least 5 of you in this room contacted me or showed me that you already did the practice test coming into the room. And I didn't even assign this till yesterday. What does that tell me? These are people that are on it. They've been on it. And by the way, these people, all 5 of them, have shown me that most of their answers were correct. Before they even came to me. They only came to me about the ones they couldn't understand. So I know at least 5 of you would have aced this test, guaranteed. A good bunch of you would have messed it up. How badly, it depends. But don't get frustrated with me when the level of effort doesn't meet the expectation. Does that make sense? The material's been presented. The material's been gone over. You've gotten all kinds of support. Many of you, I've seen only a few of you after school from 8 to 8:40. Only a few of you, like 5 this week, have sent me emails asking me to explain something. One person requested a Zoom meeting so we can go over it, which is cool. Shows motivation to me. So when you're going through this material, you have to understand, I can present it, but you have to learn it. And parents have come to me and said, well, they don't know how to learn it. Honestly, my opinion is you just don't want to. You have to make index cards. You have to memorize the material. You have to break down the notes. You have to come to tutoring if you don't understand. But when you don't come to tutoring, when you don't create note cards, when you're not verifying that the information is correct, when you're ignoring it until the day of the test, as most of you here in this room have done, because this is the day of the test, then that is not you not understanding. No. That is not it's too hard. No. That's you are not organized. That's it. Not organized or not motivated. I don't know. But whatever the case, you got to correct. Okay? You have to correct. This is only the first of many tests. All right. It's all going to be on YouTube as soon as we're done here. For the rest of your period today, I want you to make index cards based on this practice test. Do you know your subgroups? Do you know carboxyl amino groups? Do you know what they look like? Look at the study guides. I've given you multiple study guides. I want to see index cards being made today. You don't have index cards? Take a sheet of paper, rip it up into 4, or cut it or rip it into 4 sections. Use those as index cards, front and back. You need them to leave. And don't borrow any from someone else unless you're going to give them back.